Chapter 1, Section 3 discusses the scientific method. Now, depending on what book or website you may be looking at, the scientific method steps may vary in order. Our book presents it kind of in a 5-step process of observing, hypothesizing, predicting, experimenting, and communicating. Now, the key is really just to follow a logical pattern when doing a scientific experiment. Okay? So with the observing part, this generally happens first, mainly in the fact that as we go about our lives, we will see things occur or hear things and wonder, why did that happen? Okay, so that's where the scientific method process starts. From there, we would develop a hypothesis. A hypothesis is going to explain those observations and data and must be tested. Okay, so it has to be a testable statement. That's key. A way to make sure that it is a testable statement is to use an if-then statement. If I do this, then this will happen. And that's just an easy way to make sure that your observations are stated in a way that can be easily tested. Hypothesizing and predicting essentially go together. When you make your hypothesis, you're making your prediction about what you think will happen or why you think something has happened. From there, you will conduct your experiment. When you conduct your experiment, you will be collecting data. Data can be either qualitative or quantitative. Quantitative data refers to things with actual numbers associated with them. So, for example, heights or weights, things that you're going to have units with. You know, this is 100 pounds. This is 10 centimeters long. Those would be things that would be quantitative. Qualitative data are more comparative values, things like this object is heavier than this object, or it could be maybe a color description. This is a yellow product that we made, or this is a blue product that we made. So those would be qualitative forms of data. Once your data is collected and you finish your experiment, you then will look back at your hypothesis and decide if your data supports or refutes that hypothesis. Now, conducting an experiment one time does not mean that you have proved something 100% true. Your data will support it or refute what your hypothesis was. And either way, that's okay. If your data supports it, great. Maybe you want to conduct the experiment again to make sure there are no mistakes. Or if your data was refuted, then perhaps you want to try again. Maybe revise your hypothesis and try a new experiment. When you're finished with conducting your experiment and you've done it many times and gotten similar results, at that point it's time to communicate those results. When you communicate your results, it's important that you have formed some sort of theory. Now, a theory is not just a random idea of what you think happened. A theory in science is a well-tested explanation of observations. So it's something, you know, you've done this experiment many times, that you've found it to be true. Other people have been able to repeat your experiment and find it to be true. Now is the time that you can form a theory. And then we communicate those results for many purposes. One could be to make sure that we allow our fellow scientists to be able to reconduct our experiments and make sure that we did everything correctly. Perhaps we want to share information with other people so that we can work together to achieve, you know, a goal of finding an answer to some sort of problem that needs to be solved. So there's many reasons why you could communicate your results. Within an experiment, a couple other things might be possible. One is to take a sample. Sometimes in science it's very hard to conduct an experiment on an entire world. So you might only use a sample size to collect data on and then compare that to the entire population. Within your experiment, there are a couple different groups. Whenever possible, we like to conduct controlled experiments. A control group is one that is going to be our standard. That's what we will compare to. The experimental group is identical to the control group except for one factor. Okay, that one factor is going to be our independent variable. That's the one that, as a scientist, we are manipulating. The dependent variable then in our experiment is going to be the variable that responds to the independent variable. It's what we're going to be measuring in our experiment. And finally, there's something also called a standardizing variable. And standardizing variables are things that we will keep constant throughout our entire experiment. The sample here that I gave you is from your book. It talks about owls hunting and the distance that they need to be from their prey in order to catch them. So there's a control group that they use to hunt in light. With the experimental group, they have made it to hunt in the dark. So the only thing that's different between those two groups is the amount of light that they have. Okay, that's the manipulated variable. With the dependent variable, what they're measuring is the distance that it takes the owl to strike its prey. Okay, so that's what's being measured, so that's the dependent variable. What could be kept standard is that you're only using owls. We're not testing different types of birds at the same time, and that there is actually prey available. So those could be standardized variables. This is just a little summary here of the scientific method and how you don't always have to necessarily go in the same order. At some points, maybe you make your observation, you define your problem, you propose your hypothesis, you start gathering your evidence, and then your evidence is being refuted. So maybe you reject that hypothesis, but you're going to go back and you're going to try something different. So at any point throughout this experiment, you can always go back and try again.